Você perdeu o breakfast.
Não foi.
Foi bom.
Tá, o café da manhã.
Não, o coffee break.
Coffee break das 10.
E o almoço, a gente passou aqui embaixo de graça.
É, a Mari me falou, a Mari mandou mensagem. Só que eu já tava fazendo comida já. Daí não ia.
Simone, você quer deixar carregando aqui no computador? Não, tem problema não, daí já fica aqui perto também. Pode pôr aí.
This is North America.
Pode, deixa aqui perto também.
A minha eu ainda não tenho.
Second section of taxonomy of biodiversity.
Iniciou, tá? Pode ficar tranquilo, vai tocar não.
Dr. Jasko Lukink from the University of Oulu. He made it certain that I tell everyone that he's retired, not a parasitologist, but a geneticist. So he has been working he wanted me to say that he's also a parasitologist globally, which is not different from any one of us. Definitely. So Dr. Yako will also chair this section, and the title of his presentation is The Many Origins of Gyractylus Salaris, Evolution Aided by Alien Host Species, and I'll let you take care of it. Thank you very much. Can you hear me without microphone? I was supposed to be in Okay, may I get the first slide?
As pessoas estão conversando com você aqui no chat.
Não, aquilo. Você pode dar.
Ai, a pessoa que fica com o negocinho preto.
Now we are. Title, what's in the name? I borrowed. We used to be, as pilots used to be, taxonomists, always. We use names for the species, and most of the time we know exactly what we are talking about, even if we probably don't. But I would like to see species concept mostly inside. I hope you can see this. Inside from Urodactylus genus there are many species, and this is the ISM7. There are primary Imaginus salaris. This salaris is important for me because it was the reason why I was studying Urodactylus, because it made big catastrophe in Norway, and that's why it was well paid. So this salaris in ISM7 includes all the different parasites, the Salmosaur, Salmon, Ovoricus, many parasites in Ovoricus, humans, and some of them we don't know. Other species, but probably they are. So this is separated from outside by IPS quite well. This variation within is very narrow, or few nucleotides among cell lines. So it is what we are nowadays using with as species delineation. There was also talking about alien species, and alien hostages may go out, introduced to Europe, to Asia, Mexico, everywhere. And among these parasites, there are many which are also found on red coat counts. But only this salmon from North America is native to red coat. All others here are European or Asian species which have been introduced to red coat trout. They found in countries. They are not. Interestingly, salmon is not found in Europe or Asia. In Mexico. Most of you know that this grassy dog style, this mama, this is the daughter, and here is the granddaughter. They are inside of each other, and this has some influence or some correlates with the reproductive system, because probably nobody wants to mate with this granddaughter as long as she's there inside. But the daughter is already pregnant, so who fertilizes her. There are many species of Pterodactyls. This is itself an uptake tree showing different fruits in different parts of the world, and these marine environments. But most interesting for us is for Wageneri fruit, because it was studied well by Göran Malmberg in the north in Sweden long ago. It happens to contain all salmonid parasites we know so far. It's freshwater in all shores, in Europe, Asia, and it's in our homes, not in any food. It has many species, probably all the freshwater fishes in Scandinavia and Russia, why not in Siberia, have at least one species of Pterodactyl from this group. Minnows have at least 12, probably 12 species, and so on. Salaris species, someone can see there's nothing stuff. This is still one we later made from different groups. Salaris says for us also, so you later more about Pterodactylus marginus, who is parasite of Arctic charge. Salaris alien Macedonia. It's something might be more favorite. Yeah, parasites of whitefish or this someone we accidentally described somebody from Finland, which is some synonym of someone. Finally, from China, no, Vietnam, Sweden, there are many species, all there's all few recited again and share species. Some people are talking about 4 times old. all these variations within species are very narrow, and the distances between species are long. It could be used for finding yeah. The reason to study Gyrodactylus salaris was that 30 years ago, someone investigally transported Gyrodactylus with salmon. Salmon was taken from Sweden, was transported to Norway. It was bred in hatchery, it was generously distributed to all rivers. Then they got eggs from different rivers, they sent the hatchling back, and then they got this catastrophe which was killing 5 rivers, 5 populations, 5 rivers so far, completely, almost completely. This experiment was not enough for us because Russians repeated the same experiment. They transported fish from Lake Onega area, White Sea, and 92% of the production out. So this parasite is extremely lethal for salmon populations in the rivers, but only in those areas which have never had contact with it. Parasite is endemic in both areas and is causing no harm. It was described from Sweden, huge specimens that are in a hatchery. Okay, this is huge species, and it is salaris. This was not known in other places because it was not present, it was no harm. So, endemic parasite, no mortality, pre-mortem parasite, high mortality, exactly what we earlier in the section. This was reported on original hosts up until south, not remaining area. This is now we go into the field, Gyrodactylus salaris, and to learn what is Magnus from Paris, grading is output. That is here is the introflex alien CO1 from we have in Poland, Macedonia, Italy. And these are in rainbow stars. This tree shows distribution of which are living on trailing. European trails mostly have been studied in England, in Czech Republic, in Norway, in Karelia, anyway. In all places, trailings have this parasite in very low numbers, low prevalence, extremely few specimens, so that we always have problems to find them. And then they are mixed along these other lakes as well. I don't see this here a reason to separate two species. This has been the proposal of some political reasons. Some people want to have Gyrodactylus timali and Gyrodactylus salaris separated. And the reasons are legal, but not violent. Okay, the green ones here are yellow are traits which are found on rainbow trout. There are names, creative names, they are reversed. And then those are Ossetia, Italy, Romania, Cyprus, also from Finland and from Russia. You know better. These sequences are from Japan, and I don't talk more about them. But Ossetia countries are own, and most of them are unknown from the north. Okay. We also have done some multi-stellar analysis of this showing them. They are all populations are living in the easternmost localities, southernmost in Russian lakes. In Lake Onega, Lake Vladimir. It's also in the middle of Sweden in Lake Vänern, which is isolated from both by Earth. And we now go into the two plates of mitochondria are unified in a group of parasites from their localities from Redwater farms in Finland, from Lake Onega, everywhere, spread widely. But there are also populations which are salmon, in one isolated locality in Russian Karelia in Kukkosella, which is salmon, lake salmon. But these parasites here are different from others here by microcephaly, and they contain two different mitochondria. This is now first time I'm telling anybody different kinds of mitochondrial DNA. It's not big. But it happens. When they hybridized, when mitochondria is coming to the hybridization of here you see that in the middle there is several metacycles, heteroplasmic nucleotides, and in lower canal these are let's say and this difference is distributed among the Scandinavian rainbow farms, so that some traits represent this haplotype, some others, and most, actually most are heterozygotes. This hybrid is not extremely old, it's quasi it probably is 2 tens of years old, but it's kind of new. Okay. When we look later with a little bit more care, we may see that okay, in these sequences which we have been reading as one homozygous maybe we can see more indications of other, because they are illegal and we will not leave them in the first place. But when we see that most types are possible, it's not really, yeah, they are. So, friends here are hybrids. We don't know where they come. We don't know where the parental parents came. We have to guess. We need to say that they are mainly parasites from Middle Europe. So far, no candidates have been found. So the area where they can be found is very wide. And not the wild populations have not been studied. With the wild populations are there some one given in Holland, several in Norway, 3 in Poland, so one in Sweden. So this is how it looks. When we look this rainbow ground parasite, red which is found in many places is in Scandinavia. Mayday, cold or something like that. I don't know. I don't care about the local different farms in different parts of the area. And there are 14 types that are here. And all of them are heterozygotes. Permanently heterozygotes. No sign of segregation. So this rainbow trout parasite varieties has two mitochondrial haplotypes. They are drifting out, drifting to this way, that way. But all the marks are heterozygotes permanently, indicating that this clone, well say clone, is not reproduced sexually at all. But it's keeping position on rainbow trout farming industry very well. Breeding every time in transport rainbow trout in the northern Europe, you get also salads. It was called salads. Every time. Even if you get a license from the veterinarian, no Saccharomyces, you get it anyway. Sometimes it's very rare, but still. Permanently heterozygous. But we have one case where this permanently heterozygous strain has found someone love. Probably this someone was finding this clone and mating with it just in this lake with those heroin current. And after that they changed the host. They went to Solomon, Atlantic salmon, or maybe first time after this mating. This is only a small fragment of the microsatellite data in this lake. There are clones. Many clones are highly heterozygous. This one fish often contains only one clone. Mostly one, sometimes two. But some of the clones are product of reproduction by some of the clones are already homosexual. And everybody who knows population genetics understands that if this is running for a while, everything is homosexual. Everything. But interestingly, if there are two genomes only, you may calculate, you may assume that both genomes contain lethal elements, so that if they reproduce lethal homozygous combinations. If they get the third genome into this game, the number of homozygous growth is dropping red plates here represent plates on Atlantic salmon, so they can be called Eurorhynchus salaris proper from Atlantic salmon. But there are two plates. This plate is from Swedish Lake Vänern. This plate is widely distributed in the Baltic Sea area. And this Swedish microsatellites of this Swedish group are clustered just in the middle of this others. These are Russian Karelia parasites. Göta Lake Vänern parasite is just one of them, most basal of them, and indicating that in the history, the lakes in Russian Karelia nowadays, Russian Karelia, the lakes in middle Sweden, they're all connected as a very big lake, freshwater lake, and those salmon populations are relics in their lake, and they maintain the parasite from that time, probably 10,000 years ago. But even this also indicates probably this Atlantic salmon parasite is originally a hybrid between something else. We have other evidence for that. So it was a hybrid. It was probably caused by grayling. The grayling parasites joined power by utilizing sex, and they created a new parasite which was randomly adapted to salmon. Salmon was available, so it was already everything it needed was just moving the populations in the lake that one front of the glacier, different positions. So this is completely in line with the Dan Bruce. If you move most they move parasites like that. But in this case, we also see that sexual recombination seems to be connected with this. They just don't move. They recombine. They try to readapt. We have not much chance to adapt, but it's here are some children. Before understanding that they can be real at the class. Okay, back to the rainbow trout. Rainbow trout is hub of evolution by this species. We don't know much about it, but as much as we know about other species which are found in rainbow trout in sea, many times we see that they are hybrids. They are not original from local grayling or trout. They are hybrids between something mostly unknown. And rainbow trout is destroying salmon populations, but given enough time, it's destroying the salmon populations in the whole area of introduction. From Vietnam, from China, Norway. There are two rivers in Norway where this actually is rainbow trout origin. Okay, this is enough. Thank you for listening. We have time for two questions at least, right? Yeah. Oh, okay.
effects, original species. For example, if you have introduced invasive species, it may affect the person from the original species. And of course, hybridization, it was taught already, may disrupt the integrity of the host genome of the native species. Therefore, we try to can the composition of parasites communities into different fish species. They differ in feeding preferences and habitat positions in allopatrids. We suppose the differences in the allopatric localities and suppose the similar composition of parasites in sympatric localities. Moreover, we try to analyze the variability of the genes of immunity, major susceptibility to complex genes. Now, on these pictures, you have model organisms. As I already said, Paracondrostoma toxostoma is an native original species of our locality, and Chondrostoma nasus is an invasive introduced species. You can see differences in localities. You can see the differences in morphology. Mostly mouth is very important because of feeding and feeding preferences in allopatry. Now, I told the southern France. It's important to know that for native species, Orbital river is allopatry for the Paracondrostoma toxostoma native species, and Allier river is an allopatric locality for the Chondrostoma nasus as an invasive species. On the south France, near river Durance or Ardèche, these form a hybrid zone. We choose 6 sympatric localities: Le Bon, Saint-Just on the Ardèche river and on the river Le Bon, and on the Durance river 4 sympatric localities: Pont-Laragne, Avignon, Pertuis, and Manosque. Because of lack of time, I'm not going to tell you details about methods, but for molecular identification of the host, it was based not on morphology but also on a genetic analysis, which is cytochrome B gene and 41 microsatellite velocity. Also, the functional MHC genes were analyzed, their variability and expression profiles, namely genes D81 and D83. On this little table, you can see nothing, but I can show you that there are numbers of total individuals analyzed from partial localities and identified as a pure species, Chondrostoma nasus as invasive, Paracondrostoma toxostoma as native, and also hybrid. On the locality orbiter, we saw only Paracondrostoma toxostoma that confirms the allopatric character of the locality, and also for the earlier, just the Chondrostoma nasus was identified. Some of the sympatric localities were already balanced, for example, Pont-Laragne, where both pure species were detected and several hybrids. And on the other hand, for example, locality Avignon, no Paracondrostoma, no native species was found, and a little number of hybrids were detected. Now, the results about parasitism. As I told that we suppose differences in allopatry, that was true. Looking in this chart, you can see that on the invasive locality for Chondrostoma, most monogenia were present, almost 100%. On the other hand, on the allopatric locality of the native species, Paracondrostoma, mainly trematoda were found. A big difference. If you compare Pont-Laragne for the population of Chondrostoma or Paracondrostoma, big decrease in a monogenia, and increase of presence of crustacea or trematoda was found. The same for the mollusk, comparing invasive and native species, decrease of monogenia and increase of trematoda and, for example, crustacea. The proportions of other species as Acanthocephala, Testoda, Ixozoa, they're very, very low. Okay, according to the species, we can say that from exoparasite, Dactylogyrus achieved the highest abundance and prevalence in allopathic population of exotic species and also in sympatric localities called Chondrostoma. Its abundance in Chondrostoma in sympatric localities was very low. The most abundant species from the Dactylogyrus was Dactylogyrus elegans and Dactylogyrus chondrostomi, that would be chondrostomi, like it was found only on a mammoth locality. According to endoparasites, Diplostomum was the most abundant and prevalent species in both fish species and sympatric zones. Now, I prepared a comparison with abundance charts and prevalence graphs. If you compare abundance on allopathic localities, you see on the beginning there is, you can see it, but here is Dactylogyrus, Monogenia here, endoparasites here. Yeah? For example, Dactylogyrus elegans, very high abundance, and in the allopathic locality, very high abundance of Diplostomum or Polyhisco. No. Oh. Something. Yep. Really, sorry. Okay. Sanjus. On the upper level, you have an invasive species, and here is native species. Chondrostoma, Parachondrostoma. Yes, you can see differences. This is the first sympatric locality, and you can see that also Dactylogyrus was found in this sympatric locality, but here is the very low abundance of 2, comparing the Sanjus where is the 10. Manos is the same, low localities for Monogenia, and no or low abundance of endoparasites in other species. Pontuara I compared also, and Avignon and Pertuis I compared because the same river locality. About the same. Very, here is interesting data. Here, very species richness in this locality. The same comparing in prevalence. Earlier, almost 100% for Dactylogyrus elegans and Dactylogyrus pistolae. On the other hand, native species Diplostomum and Polyhistomum, yeah. Sanjus also prevalence, not just 100%, 60 of the Monogenia here. Manos is the same, a big species variability. Okay, that's now, comparing the similarity in parasite communities, we found the lowest similarity in allopathic localities according based on parasite abundance or parasite presence or absence data. For example, significant differences were found in similarity in pairs of population, allopathic chondrostoma, sympatric chondrostoma. Allopathic parachondrostoma, allopathic sympatric toxostoma. When we take into account the parasite presence or absence data, no significant differences in similarity were found in a sympatry or in a between population chondrostoma, parachondrostoma. Now, we found, according to hybrids in this locality, hybrids between these two species present a lower parasite, exoparasite presence than invasive species. Here, chondrostoma mesus as invasive and parachondrostoma as native comparing with hybrid. The same result we were found when comparing bacillovirus only or monogenous group only. In the endoparasite, the hybrid tended to have a fewer endoparasites than pure species. So, the first chart I showed you was the analysis of the identification of hosts, that it is pure species or whether it is hybrid. We say do this based on the variability of MHC genes, and you can see that allopatric character of the localities for pure species was confirmed. But you see the gene flow here. These mixed parts indicate presence of hybrids, and according MHC genes analysis, you can see the big gene flow, both directional, in also in an allopatric locality. Yeah, so they are very much more precise than analysis of microspectral, according to gene flow. On the chart, I just wanted to compare the number of individuals in each locality. That means that big black line and the red line is just like alleles identified. You can see that it aligns with the number of hosts. What is interesting on this is that DAB1 gene is in this blue line, and the allele of DAB3 gene are in a yellow line. You can see how many alleles in each locality has the DAB3 gene, and what was the lower number of alleles for DAB1 gene. It's very important for our results. According to specimens, how many alleles have a specimen? Here you can see that for native species, they prefer to express these genes only mostly in two allelic profile. On the other hand, when invasive species have 3, 4, and more alleles. It is very important and nice is that the hybrids have the intermediate profile of expression of these genes. So, what it can say? First of all, if you have alleles expression profiles for DAB1, it's on the right or left on your side. You can see that many individuals did not express DAB1 gene. On the other hand, invasive species prefer to express this gene only with one allele. And DAB3 lineage, except earlier or here as the allopatric localities or PEP3 as sympatric localities, all specimens express the DAB3 gene with the 2 or more alleles. What can I say? In sympatric areas, we have more diversified DAB1 gene than in native species than in the invasive species. That means that, and invasive species, on the other hand, whether if they have a lower number of alleles, they prefer to express more both DAB lineage and more elements per specimen, and this can have an influence on the homogeneous part of the flow. We should also confirm it. Nice.
Actually,
No more? Okay. So I would like to thank you for listening me, and
thank you.
we can discuss in more.
So much information that it takes few minutes to digest. One question. It's very interesting.
Good afternoon. I'm talking about Geodactylus. Do ladinho lá. Não, aqui é dentro. Geodactylus is a genus that possesses more than 400 species, so high genetic and morphological diversity. However, it is not considered a natural group. Still, species have available molecular data, and most of them can only be found in museums. Morphological diagnosis is essential to the establishment of a natural classification of the Geodactylus, and therefore it is necessary to evaluate the use of molecular and morphological techniques to delimit the species and suggest phylogenetic hypotheses. So we use geometric morphometry to test if morphological characters can be used to define natural groups. Geometric morphometry analyzes morphological variability between different species. Homologous regions can be used for the delimitation of the form and the structure. Okay, in the study, we used a total of the 27 species from existing molecular phylogeny. The images were taken from published papers and books. The hook is located in the ector regions, and the use for fixation on the host and the structure to obtain the data. All pictures have been compressed into program TPS into, and points were scored in the program. Landmarks are located in homologous regions between species. We'll see 3 points in each hook. The point 1, point region, and point 2 are the insertion of the shaft and the base of the hook, and point 3, the toe. The pseudo landmark used to define the shape of the hook were also recorded in the image, with 25 points between point 1 and point 2, 50 points between point 2 and point 3, and 25 points above the point 3 to point 1. And each image was analyzed 3 times to avoid mistake of numeric points, and the data were analyzed with morphogenesis. The first analysis performed for crusts with removed variation in size, rotation and scale of the image, maintaining the shape. In this graph, you see the blue dots. They're indicated the more accumulation of the points, the black points. There are all markings made for all species. Also, you observe the grade variation in the analyzed species, such as the location of the site of them. An important step for this analysis are the boundaries of the classifiers. For example, when used one to distinguish variation between populations, which population should have a particular code. And in these study classifiers were the largest monophyletic groups from Gyrodactylus. As the molecular phylogeny was already known and was divided into 5 groups: Group A, Group B, C, D, and Group E. To be the classifiers, which group contain closely related species for which the design of the book was available. The principal component analysis aims to eliminate overlap and choose the most representative data. Principal components 1 and 2 cover 65% of that variation. In this graph, you see that closely related species tend to be close together, what means that they have a similar shape. In this case of the group D. And group A and E are also phylogenetic and morphologically close, but PCR codes do not separate these groups. The canonical analysis uses linear combinations to analyze the similarity and similarity of the groups in the data set, find the way to best distinguish the groups of samples. In this analysis, you observe that there is a great similarity between morphological and molecular data. Morphological groups well separated according to the molecular phylogeny, except point. This point is the group E, but is more close the group A. And this point is Gyrodactylus salax. Our results suggest that focal morphometry is a good proxy for monophyletic groups related, revealed by molecular systematic of Gyrodactylus. Thus, the result of the study strongly indicated that the division of the Gyrodactylus based on the recognition of monophyletic groups combined with geometric morphometry allows the inclusion of species for right DNA sequences non-available. Future studies should take into account other structures, such as anchor and bar, to give more support to this procedure. So, thanks.
Did you run the image analysis on the drawings or on the live images of the slides?
I'm sorry, I don't.
Sorry. Did you run the image analysis on the drawings or the hooks or on the live images of the slides, for example?
Se você usou espécimes de fotografias ou desenhos de trabalhos publicados ou sobre os animais publicados?
Todos foram desenhos de artigos publicados. Images of the papers. And I scanned the images and I used the program. Images of the papers. Images.
Drawings. Okay.
Did you test how the correct shape transformed by your method to the computer is dependent on the number of points? Because I think that the different number of points must be very important for the result.
How many numbers? Ele só perguntou se você usou números diferentes para testar a resolução. Se aumentando o número
De número do
número de
landmark. Yeah, but it's similar. I use more points because because I feel better out form of the hook. So I use much points. So 25 points. Yes, the same number. Same number.
I think that to be precise, you must use the same number of points in all cases, in all species, because
Yeah, the same number. The same.
I would like to ask, unlike the drawings from the publication, was it the first time that you corrected or tried all the publications? My experience from all the publications is that the drawings is definitely precise. Because the presentation of this data, so then you access data that are not really precise. How many publications was from the last decade and how many publications used when you analyze?
Quer saber quais são os trabalhos, quantos trabalhos recentes, quantos trabalhos antigos?
I don't remember, but in the papers on sheets. Old papers.
Some of it's new.
But in these future students, I have worked with the slides of the museums, the slides of the
Slide sets.
because the error with the image is very big.
The images are good, so you use the slides.
Yeah, because they like in the future.
I didn't like the idea that the oldest the drawing is or the oldest the work is, the worse it is, because we're getting old.
Very interesting for us because everybody wants to make some money from all these collections. In my generation, it's the sheets. I agree. Okay, next.
So, more questions?
Hello, I'm Fan Xie. I come from China. This is the second time I come to ISM. Today I will introduce the new features of polychroma from runner tutorism. in China. There are some pictures about the new polystroma by micro. And then I will mention about the background of Chinese polystroma. The family polystromatic is composed of 24 general more than 2,000 features distributed globally, but little is known about polystromas of China. Only genus Polystroma data and the genus Diplopus of that has been recognized. And until now, representing 12 features is the first feature named in 1936. Now, I give the list of the parasite, host, and the author. You can see there are 6 Diplopus in the list. There are 6 polystomas in the list. The first Diplopus is named by Li Luoying in 1936, and the first polystoma named by Zhang Shuyi and Lang So in 1987. So there are 12 polystomatids in China now. These features were published by the Chinese publication after the taxonomical technique. I will mention some features I reported in the last system. The first is Diplopus chelicans, is named by my team. This feature is smallest body size, can separate it from recorded polystoma of genus Diplopus. That frog is the first. It's very popular in my country and in my province. The second is parasitic in Rana grahami. This parasite is Diplopus grahami. The body size is similar to recorded features, but the head tone is very smaller. This can separate it from the recorded features in genus Diplopus. And in one host, we can find more than 60 parasites hovered in the urinary bladder. And then this parasite I reported in ISM7. The host is rhinomosin. We got the host from Vietnam, near the Yunnan province. And this is the reproduction system of the parasite. The body is elongate, and the intestinal is very simple. The second part is the polystoma. I described the first polystoma is polystoma pimpinidens. Pimpin is a county in our province. It is very fascinating. We found more than 10 features in the county. These polystomas parasite in the host security, and we described it because the outside layer is intestinal, completely absent. More than 13 or 14 major developed branch. So we better named this as a new species. And my second polystoma is parasitic in Dutch frogs. And this parasite major intestinal vesicular branch and extend well beyond the midline, form the very confused canals. And the new speaker is named by my graduated student, Zhao. She graduated from our school. He named a new species named Polystoma pimpinidens. We found more than 6 new species undescribed from Xinjiang County. This new species parasite in Hala. And we can find the very simply instant and very different formulae and very different the circle in this parasite, and the body size is the smallest in China. This is a new polystroma named after my supervisor mentioned, Professor Zhou Yanjie, and Mr. Master degree supervisor, polystroma zoe. Out of 73 frogs, 4 male hosts were infected. This new species is smaller than the report species, but the size of the hectare and the humula is different. It's larger than the report species. Now I will report the new species polystroma from Rana Chuchunism. This is the location. We found the polystroma in a country named Lufeng. It's middle, it's central area of Yunnan province. Yunnan province is in the southwest of China. It's nearly to Vietnam and Thailand. The Thai coast is Yunnan Churches. The site is mineral black. The frogs were found along rivers in open grassland forest. Originally, the advent started in December, but peaked in June. It's very special. Most of the frogs were laying eggs in summer days, but the Rana Chuchunism laying eggs start in December and in June. So we found the frogs in the near the December and near the January. We get the frogs from this plant in water. They pick this get warmer in the cold water because it's winter now. The temperature is nearly 0. So we get the host by hand. This is my sister just about 10 years ago. So I get older now. We get the frogs from 2002 until 2009, and we detected 578 frogs. Somebody curious asked me why can you detect so much frogs? And I give the answer like this. From 2002 and until 2005, we get the different polystomas from the same host. This is a smaller one. This is a new feature I reported. So we get the morphological about these two polystomas, except the size of the body. There are difference between them. The hamuli and the silk and this is very different. So we think these are two different morphotypes. But are they two species? Are they cross-infection? This is the problem. But I'm so sorry. We just get the different forms for twice. In 2002 and in 2005. Until now, we get no new materials about this interesting question. So, this is the new species picture and the measurement. The measurement based on black parasite treatment is 13. This feature both using reflected with two branches during the process to form a hectole, exciting well into the hectole, and 6 or 7 major internal vesicular higher branches to form a network. This is very special, and the lateral vesicular joined to form a peripheral network, and the hectole circle is large. There is the hamulized marginal hoofless picture. And there is the genital bubble and the genital span. It's very special. I will discuss with Professor Louis Duplessis, a very promotion and covers most of the original posterior to the original. This is the thesis of the neoplasma. Under the microscope, the thesis is follicular, made of specimens above between the testicular follicles. And we discussed the new species is very similar or closest relative these two species recorded. The one is the first polystroma integrarium and the polystroma parvarium. The new species is distinguished from the two by an uniform round ovary here and very large testes. Although both of them present distinctly branched in skin. Conclusion by morphological reorganization combination of characters. Firstly, the distinct formally dense with cartilage network, as wider and oval-shaped ovule, and the third a very prominent and covers most of the testes with region posterior to the ovule, and the fourth, the shape and size of filter, commuline and marginal hopefully. What's the relationship between the relative features? Sorry.
No, it's 12 minutes. Just time. 12 minutes.
I'm sorry. Okay.
12 minutes.
We do some molecular research about the relative features. We can say, based on ATR engines, we found that the three species are in the different branch in this picture. The new species combination a single branch, and the reported species are different terms. So MLT based on partition synthesis of AGSR gene can tell us maybe they are different species. These research are priced in the publication. And conclusion of molecular research. Molecular research is a phylogenetic relationship based on MLT from partial AGSR gene sequences indicate that the species is well separated from its close relatives, such as Polystoma intervarium and Polystoma cavula. This supplemental molecular research suggests that under described the Polystoma should be considered as a new species. As a final acknowledgement, this study was supported by the National Nature Science Foundation of China and the National Science Foundation of Yunnan Province. And our special appreciations to Professor Claude Combs for identifying cancer and providing references. And this article is handed to Louis and my supervisor of PhD, Wan Xu professor. I come from School of Life Science in University, China. Also, the future, after my two graduate students graduated, so my team just have 4 people. So just as Louis said, we should develop the broad research team. So thank you for your attention. I'm confused for the different times, so if you have any question, we will talking after the meeting, or you will ask me. Thank you. Thank you.
impact program because it helps us to provide many supplements to applications, and this is therefore organize this one more important. We have to exchange our research progress each other. So I'm from China, South China Northern University, Guangzhou. Probably some of you visited this city because ISM7 5 has been here in this city. Today my presentation is about propose a new genus and two new species of Mesocritic quite 1933. Here I briefly introduce about this species. This species, they have two pair of clams on the capture, and the clam type is Mesocritic type. According to Mama Ye's work, this clam consists different skeletons. Here I show you such as SIL, SIS, SMB, and in different genus or different species, the skeleton may be different in different genuses. But so far, all species in this family have this type of clam. This is some background. And we search homogeneous from a marine fish. One of the fish is cloaked fish, this one. And this fish distributed, this is the area show the description of this fish. Actually, this is image from download from this website. This is South Channel Sea here. This is South Channel Sea. And we search homogeneous from this fish and near this site. 大亚 Bay. Yes, we get different species, but we found some is different. Here I show you this picture show you the hectare. This is the hectare of the one species. We notice besides this normal Mesocritic class here, three pairs of them. This three pairs. We notice there are different clams here, different kind of clams. In order to identify the skeleton, we separate this clam and digest with enzyme called cascade, and we try to separate this skeleton and found the structure is totally different from a long Mesocritic type. We named it long Mesocritic type. Actually, they consist of three kinds of skeleton. One we named is SCU, this one. The other is outside one. The shape is like this. And these two is internal one. They have a different shape when we observe from different angle. So this is the first time this long Mesocritic type plant was observed in this family. So I think it should be a new genus in this family. So we named it Mesocritic new genus because the form is little like a pair of the of this Yeah. And we named one of them is type species. Actually, there are two kinds of new species in this new genus. When we compare them, we found they're different. The difference in shape. This is a little slim, and this one is like especially when the womb is living. So we named it as dead cross, means it's like a bottle, a bottle. And the size is quite different. We need this size, length, and the wide is different. Actually, this is much bigger than this one. Besides this size, we can see the hectare. This species, the hectare is clearly separated from the main body, but it is not so clearly. And I mentioned the type, the other three pairs class is membrane type, but when we study the details skeleton, we found they are different. The structure is quite different. We separate this skeleton as well and found the composition, the component is different. For this snail species, they consist of 5 skeleton, but they have this XAA structure and this XL structure, but for another species, they have this SPS and SAP. So the skeleton is different in these two new species. Besides the different structure of skelet, we found the structure of the male's terminal structure, actually the copulatory structure, is different. Is a big difference. For this male this species, we found their spine on this male's copulatory organ, but the number of the spine is different between these two species. There are less spines and there are more spines. And besides out spines, they have 2 pairs of spines, but it's only 1 pair of spines. So they're truly different. Besides, and the hector they have, and the terminal of the hector, there are 3 pairs of this tabula on this species, but only 2 pairs of tabula on this new species. And the last one, the Foxsocker, is nearly the mouse. The structure is also different. They have 2 separate sockets, but for this species, the socket is similar and only one. They're not greatly separated each other. So for summary, we take up new genus and identify 2 new species. One is now, actually this species named after one of most famous scientists, Professor Liang. He passed away 2 years ago. And this another species. Actually, this work was submitted to systematical parasitology and then conservation just now. We probably after this completion, we will submit the revised version. Although this Melocrine family, although it's set up as early as 1792, but at this moment, it's totally 25 genus was reported in this family. And we summarized
we summarize the class arrangement on the hectare. Usually, they have 4 pairs of class on the hectare, and the arrangement for most genus is arranged bilaterally symmetry. So that is both sides have a same number with the same size. So most genesis is this form, but there are some variation. For example, for this pseudo as a colloids, one pair of clasps is much bigger, and for this quarterly, the 2 pair of them are much bigger, but the structure is similar. The structure is similar. We list this new genus as this because they have a very unique class. This is quite different from all the species in this family. In addition to this type, they have another one. That's the arrangement of class is not same on both sides. For example, this heterocotta, there are 2 large clades on one side, and the structure of this big clade is different from the small one. We still have another genesis, Samuel Meadow Crest, in 2003, because they have 3 large cats on one side. Actually, there are other different, other 2 genus, they have the class different from both sides. But so far, there's still, so we're wondering if this genesis exists that is on this side, only one big class exists. We still don't know, but we propose probably this have this possibility. In mainland China, totally 12 genesis in this family was reported. Actually, this work was supported by National Natural Science Foundation of China to Xue Juan. She will present her work later. And we would like to expect David for many literature. And we also thank Jin Zhang, our supervisor to a master's degree. Although Professor Zhang is retired, he is working on a huge work in writing this book, Follow Silicon Technology Part 2. And probably this work will be finished in 2014. And probably next autumn we have a chance to see his work. And this is our research group on monitoring. Professor Zhang is here so happy. Why? Xue me another body tells us actually Xue Jian's and the work was from Chinese Han Yuan. He graduated from our university. We go to another university, then ISM University for his PhD study. Okay, that's the presentation. Thanks for your attention.
How much is this whole time of this new
course, we collect the parasite on you. But when we vaccinate, we put some solution on protein K. The protein K. Protein K. Yeah.
Good.
Yeah.
I don't know very much. You mentioned the heterocultivate at the end. You mentioned that even heterocultivate unital, but thanks to the power of the internet, I have checked and heterocultivate unital is synonym of heterocultivate monocultivate. And this I found in the database by Dominguez. Just a reason.
Good afternoon, everyone. I talk today on enterovirus disease from South Africa, found from the southern north to the 011 London. African species of the family Cichlidae are classified by 5 genera of monogenous that belong to the family Dactylogyridae. And among these, we have 3 gill parasites, namely the highly identified Pseudogyrus species and also Onchopdella Pseudogyrus. We also have 2 endoparasitic monogenous that infect cichlids, and these are Urovirus that infect the urinary tract of cichlids. We also have Enterovirus that infects the stomach of cichlids. To date, there are 12 known species of Enterovirus, and previous records are from Mexico, Africa. We also have from Germany and Israel, as well as Sri Lanka and India. And some of the records are a result of introduction of books. Previous records are mostly African, and namely from Senegal, Ivory Coast, Cameroon, and Egypt. Focusing on Southern Africa, we have only 1 record of enterovirus, and this was recorded from the Tawa River, which is part of the Greater Limpopo River system in South Africa. Parasitological surveys were carried out by the University of Johannesburg Parasitology Group, and these surveys were carried out in the vicinity of University of Johannesburg in Pada Dam. This dam is part of the West Dean tributary system of the Greater Limpopo River system as well. During these parasitological surveys, we encountered an endoparasitic monogenian of the genus Enterovirus, and this was in the stomach of the southern mouth brooder, Pseudocranilephus filanda. In the field, fish were collected by maintenance and they were killed. The gastrointestinal tract was dissected and parasites that were encountered were carefully detached from the stomach mucosa. And in the lab, specimens were mounted on a glass slide using a mixture of gas. The prepared slides were also filled with clear nail varnish. Observations, drawings, and all measurements were done using the light microscope. Preparation for SAM, specimens were dehydrated in a graded series of ethanol, subsequently in a graded series of hexamethyldesilazane, and they were sputter coated in gold and then examined. The dimensions of the structures, the structural structures that were used in this study were followed those of Buzel, and as adapted to enterovirus by belong. The nomenclature follows that of Parizel and Buzel, where they used ancinnulus for small marginal booklets, and the grippers for hamulus or anchor. The main structural structures for enterogyrus are the dorsal grippers, the ventral grippers, the ventral transverse bar, as well as the uncinulus. And for these, the dimensions that we used are as follows: the anchor length, the blade, the shaft, the guard, the point. And for the ventral transverse bar, the length as well as the width were taken into consideration. The uncinulus also, we looked at the length. Enterogyrus specimens were actually seen anchored firmly to the stomach mucosa, and in some specimens, in some stomachs, we could actually see some eggs being released from the anterior part of the parasite. The parasite has two pairs of ocelli. The anterior part is smaller and wider spaced, whilst the posterior part is larger. It is situated closer to each other, and in some specimens, the posterior pair was actually fused on the median plane. It is covered by a thick tegument, and posterior to the ocelli, we had a large rounded muscular ventral pharynx. The parasite is pear-shaped. It has a pear-shaped body which extends posteriorly, and this is where we find the hafta. In some of the specimens, the uncinula were seen to surround or to encircle the grippi, the transverse bar, as well as the first pair of uncinuli, whereas in other specimens, we had two pedunculae, the anterior peduncula, on which we had 6 pairs of uncinuli, and on the posterior peduncula, we had 2 grippi, the ventral and the dorsal, as well as the ventral transverse bar, and the first pair of uncinuli. This is what we saw when we observed ISM, the uncinuli and the ventral grippi. A closer look at the ventral grippi and the dorsal grippus. The dorsal grippus has a bifurcate root, and the blade is caved. The ventral grippus, when looking at the measurements, was smaller than the dorsal grippus. The ventral transverse bar is thin. It is slightly sclerotized. It was very difficult to see using the light microscope, as well as thin. The uncinuli are also slightly sclerotized. The morphology of the reproductive system is typical for the genus, and the serous is situated medially in the anterior quarter of the body. It is slightly sclerotized, it is tubular, it also forms a base that tapers distally, and it forms a continuous spiral, as in other species of the genus. Based on the measurements of the haftoral structures, the specimens in the present study were quite similar to those of E. melanensis and E. barombiensis. These 2 species were found in Cameroon, and they were found in different species of cichlids. However, based on the spirality of the serous, the specimens in the present study were not similar to the 2. We had, just before we came to Brazil, the day before we left, we got some sequences back, which we had sent to another university. And the sequence of the specimens, based on the C1 and D2, show that they are closely related to E. coronatus, which was described by Parisot from Senegal. And so we need to look at the analysis so that we can determine whether this constitutes a new species or not. Thank you.
Yeah. What you said is the one I was so certain.
I can't remember offhand, but
compare the sequence of this new, probably new species with only Antellogeus coronatus, or you have other sequence for other species of Antellogeus?
The only sequences that we could get were coronatus and Enterogaris species 1, as well as Enterogaris species 2. And I think this was from one of your papers. Yeah. So there were 3 sequences of Enterogaris so far. From many local, yeah.
So it's close to coronatus?
It's closer to
How much
coronatus.
difference you have? I mean,
Only 2 best pairs were different.
For your scanning and chemical microscopy, how did you treat the specimens? How did you prepare them? Did you clean them up? Did you try to clean them up or not?
The specimens are so tiny, if you try to tamper around with them, they can actually break. So we didn't really clean them.
Because I noticed that you have a problem with mucus.
Yes.
Something you can maybe think about is to use contact lens solution.
Okay.
The usual solution of contact lens solution. You just put it in that, shake it a bit, and then fix it. So don't fix before you clean. And you can really clean it.
Thank you.
we may put them after in a sonic, ultrasonic. Yeah, it works pretty well.
Okay, thank you.
Okay.
Hello, everyone. My name is Chinese called Ding Xuejuan. Ding is my family name. Xuejuan is my best name. I'm focused on the technological studies of the polystromatism monocylia from annulus in the South China. Just like Li Xian and Ruiz mentioned, there are about 200 polystroma species are now. But before 2006, only 9 species have been recorded in China. That's the species, or the 9 species recorded before 2006. And it's distributed in the this is the map of China. The Yunnan is Li Xian just mentioned. The Taiwan, 2 species of turtles recorded in 1930s. Another one is in the Hanker Lake between China and Russia, and the other species in Beijing, Jiangsu, and Zhejiang. So in the South China, including Guangdong, Guangxi, and Hainan, no any species have recorded. And the National Nature Science Foundation, we studied investigated the monogenia from amphibians and babies. Just in the South China area. The location we investigated in the Hainan too is the Jianfengli. That's the Wuzan in the Guangdong is Honghuangshan, Linnan, Gudousan, Shimentai, and Guangzhou. 广西的10万大山和大凉山。Sorry. His name is called total 54 species of amphibians and 7 species of charonians were checked, and 8 species of Polystoma monogeneous were collected. The hospital we investigated about such as the rhino. And this small progress we put in the photo, and they are put legs. When we collect the legs and find that there are polystoma legs, so we designate and find that they are in that monogeneous. That's the plan of the progress in the monogeneous. 6 shots. The walls are filled of the eggs and very this is the seal. Those specimens filled of eggs are collected in the spring, in the April, and these specimens are collected in the autumn, in the September and October. Then little eggs and small eggs in the body. That's eggs collected. The larva developed in the egg and outside. That's the specimens. The 8 specimens as we collected are the first species, the second species. And ISM we were studying. The hosta are very rare in the water. It's just found the second hosta, the broadest in China and in the world. So all the 8 specimens we found are new reported in the South China, and 1 species and the genus are new reported in China. Their hosta belong to the 4th genus. There are no polystoma has been found from the channels and the salamanders in South China here today. That's the species we collected, and 3 of them are new species. After 2006, Li Xian have described 3 new species from Yunnan province. So up to now, there are 17 species Polysperma monogenealis only in China. To compare this, the number is small in one country, but there are recorded more than 300 amphibians and 37 locals that are in China. So we think there are many works to do. Just students of groups and take part in this work. This is the routine. Many of them, this Professor Zhang with me, and all of these are students of Professor Zhang. Many of us we are working on the monogeneous of the others. That's okay. Thank you.
I'm so excited. My dad used to put this like this. A question that I have. The part of this that you found on what basis did you decide it was part of this? Yeah, but which characters did you use to identify? You sure it's not pseudopolis? Because parapolis has got multiple pieces. We can discuss afterwards. Thank you.
Yes.
Are there more specialist questions? Sometimes I think that being a tenant and for okay. Now we're coming back to narrow points and applause.
Representem de South America Monogeneia, para as artofícios, anfíbios e répteis, de Simone Cohen Márcia Justo. South America is bordered on the north by the Caribbean Sea, on the northeast and east by the Atlantic Ocean, on the west by the Pacific Ocean, on the south by the Antarctic Ocean. The only countries that doesn't present a sea coast are Bolivia and Paraguay. On the northeast and east, we have Colombia, Venezuela, Guyana, Suriname, French Guiana, Brazil, Uruguay, and Argentina. On the Pacific Ocean, on the Pacific coast, we have Chile, Peru, and Ecuador. Among the island possessions, we have the Galapagos Islands on the Pacific Ocean and the Falkland Islands on the Atlantic Ocean. The first species of Monogenoidea described from South America was Benedena hendorfi in 1889 from Chile. The second species that Marcus showed already was Pidericanella ovipula in 1894 from Brazil. And then Lophocotyle cyclophora in 1896 also from Chile. Only in 1938, more species have been referred in South America by Price from Brazil and by Mazet from the Galapagos Island and the neighboring Pacific. From 1944 to 1962, more species have been referred from Chile, Colombia, and Uruguay. But only from 1965, the knowledge of the South American Monogenoidea developed, mainly from the species described from Brazil. Considering the new species described and others that were considered synonyms, 651 species are referred till now in South America. From Brazil, we have 437 species from 27 families and 134 genera. Argentina, we have 75 species from 12 families. Chile presents 40 species from 27 genera and 12 families. Uruguay, 17 species from 9 families. Paraguay, only 4 species with 2 genera from 2 families, Dactylogenidae and Polystomatidae. Bolivia, 12 species for only the family Dactylogenidae. Peru, from Peru, were described 114 species from 21 families. Ecuador, 7 species from 3 families. Colombia, 17 species from 5 families and 10 genera. Venezuela presents 44 species from 16 families. Guyana, 4 species from 2 genera and 2 families, Polystomatidae and Dactylogenidae. Suriname, only 2 species from Thoracocotylidae. French Guiana, 6 species from 3 families and 4 genera, but today we have noticed that Lewis found several species of the family Polystomatidae that he will publish very soon, we hope. From Galapagos, we have 11 species from 8 families, and from southern Patagonia, 23 species from 9 families. These numbers do not represent the real Monogenoidea fauna of each country, but only the number of species reported presently in each geographical area by the few researchers who have worked in this region. This catalog updates the papers published in 1998, in 2000, and in 2008. And we dedicated this work in memory of Professor Yolanda Perna, with whom we worked with him. We started our studies in Monogenea in 1963 in Hadassah Medical School in Israel 50 years ago. So now, we edited this book to celebrate the 7th International Symposium Monogenia and the first meeting in South America. And we have the pleasure to offer it to all our colleagues that are interested in it and can receive it during the coffee break and during the post-event.
Thank you very much. Biodiversity is probably the highest in the world